A constant source of blood glucose is an absolute requirement for human life. That is why the body developed mechanisms for storing a supply of glucose in a rapidly mobilizable form, namely glycogen. These mechanisms are glycogenesis, the conversion of glucose to glycogen for storage, and glycogenolysis, the conversion of glycogen back to glucose for utilization. Let's first discuss glycogenesis, which is composed of 4 steps. Step 1. The synthesis of uridine diphosphate glucose. It is synthesized from glucose 1-phosphate and uridine triphosphate acted upon by the enzyme UDP glucose pyrophosphate, forming now your uridine diphosphate glucose. Step 2. Synthesis of a primer to initiate glycogen synthesis. Since adding UDP glucose and other glucose molecules to elongate the chain via glycogen synthesis cannot occur, a primer should be first formed. A protein called glycogenine can serve as an acceptor of glucose residues from UDP glucose. Glycogenine itself can catalyze this reaction because it is an enzyme. Just add a few more molecules of glucose from UDP glucose, producing a short alpha 1,4 linked glucosyl chain that can serve as a primer that is able to be elongated by glycogen synthase. Step 3. The elongation of glycogen chains. You can now add molecules of glucose from UDP glucose at the non-reducing end to elongate the chain via glycogen synthase. It is the enzyme responsible for making the alpha 1,4 linkages in glycogen. Step 4. The formation of branches. The branches are made by the action of the enzyme amylo alpha 1,4 to alpha 1,6 transglucosidase. It removes a set of 6 to 8 glucosyl residue from the non-reducing end of the glycogen chain, breaking the alpha 1,4 bond and attaches it to the non-terminal glucosyl residue by an alpha 1,6 linkage, resulting to a new non-reducing end. You can now repeat step 3 and 4 to form a highly branched polysaccharide named glycogen. Now let's go to glycogenolysis, which is composed of 3 steps. Step 1. Shortening of chains. The enzyme glycogen phosphorylase cleaves alpha 1,4 glycosidic bonds between glucosyl residues at non-reducing ends until 4 glucosyl units remain on each chain before a branch point. The remaining structure is called limit dextrin, and phosphorylase cannot degrade it any further. Step 2. Removal of branches. The branches are removed by the debranching enzyme. First, it removes the outer 3 of the 4 glucosyl residue attached at a branch. Next, it transfers them to a non-reducing end of another chain, increasing its length. The remaining glucose residue attached in an alpha 1,6 linkage is still removed by the debranching enzyme, releasing it as free glucose. This glucose chain is now available again for degradation via enzyme glycogen phosphorylase. Step 3. The conversion of glucose 1-phosphate to glucose 6-phosphate. Since the glucose released by the glycogen phosphorylase is still in the glucose 1-phosphate form, it has to be converted first to glucose 6-phosphate via enzyme phosphoglucomutase. In the liver, glucose 6-phosphate is transported into the endoplasmic reticulum by glucose 6-phosphate translocase. Here, the glucose 6-phosphate is converted into glucose via glucose 6-phosphate. Hepatocytes release this glycogen derive glucose into the blood to help maintain blood glucose levels. However, in the muscles, glucose 6-phosphate cannot be sent into the blood because of the lack in the enzyme glucose 6-phosphatase. Instead, it enters glycolysis, providing the energy needed for muscle contraction.
The pentose phosphate pathway or PPP branches from glucose 6-phosphate produces NADPH and ribose 5-phosphate and shunts carbons back to the glycolytic or gluconeogenic pathway. The PPP has been demonstrated to be a major regulator for cellular reduction, oxidation, homeostasis, and biosynthesis. Every living organism has a set of blueprints in each of their cells called DNA and RNA. These blueprints are initial for life because they are the information on how to build the proteinic structures that make up each and every one of us. Given the structural and functional importance of DNA and RNA for all living things, there are many layers of quality control to help avoid and correct mistakes when DNA and RNA are initially made. While the products of glycolysis are sent through the rest of cellular respiration to produce energy, there is also an alternative branch of glycolysis to produce the sugars that make up DNA and RNA. This pathway, called the pentose phosphate pathway, is special because no energy in the form of AATP or adenosine triphosphate is produced or used up in this pathway. How does it happen? Similarly to some of the processes in cellular respiration, the molecules that go through the pentose phosphate pathway are mostly made of carbon. The easiest way to understand this pathway is to follow the carbon. The breakdown of the simple sugar glucose in glycolysis provides the first 6 carbon molecule required for the pentose phosphate pathway. During the first step of glycolysis, glucose is transformed by the addition of phosphate group, generating glucose 6-phosphate, another 6 carbon molecule. The pentose phosphate pathway can use any available molecules of glucose 6-phosphate, whether they are produced by glycolysis or other methods. Now, we are ready to enter the first 2 phases of pentose phosphate pathway, which are the oxidative phase and the non-oxidative phase. The oxidative word of this phase comes from the process of oxidation. Oxidation is breakdown of molecule as it loses at least one of its electrons. This phase is made of 2 irreversible steps. Step 1. Glucose 6-phosphate is oxidized to form lactone and ADPH is produced as a byproduct of this reaction as star superscript plus and superscript is reduced as glucose 6-phosphate is oxidized. Following the oxidation of glucose 6-phosphate, another reaction catalyzed by a different enzyme uses water to form 6-phosphogluconate, the linear product. Step 2. Next, a carbon is removed and CO2. It starts subscript 2 and subscript is released. Once again, the electron released from this cleavage is used to reduce start superscript plus and superscript to NADPH. This new 5-carbon molecule is called ribose 5-phosphate. The non-oxidative phase is really handy because these reactions are reversible. This allows different molecules to enter the pentose phosphate pathway in different areas of the non-oxidative phase and be formed up until the first molecule of non-oxidative phase. Ribose 5-phosphate is the precursor to the sugar that makes up DNA and RNA and is also a product of oxidative stage. Step 3. Ribose 5-phosphate can be converted into 2 different 5-carbon molecules. One is the sugar used to make up DNA and RNA called ribose 5-phosphate, and this is the molecule we will focus on. Ribose 5-phosphate isn't being divided because the carbon count is the same in the next step. Step 4. The rest of the cycle is now made up different options that depend the cell's need. The ribose 5-phosphate from step 3 is combined with another molecule of ribose 5-phosphate to make 1. 10-carbon molecule excess ribose 5-phosphate, which may not be needed for nucleotide biosynthesis, is converted into other sugars that can be used by the cell for metabolism. The 10-carbon molecule is converted to create the 3-carbon molecule and 7-carbon molecule. The 3-carbon product can be shipped over the glycolysis if needed. That being said, recall that we can also work our way back up to another molecule in this space, so the 3-carbon molecules would also be shipped over from glycolysis and transformed into ribose 5-phosphate for DNA and RNA production. Step 5. The 3-carbon molecule and 7-carbon molecule from the interconversion above in step 4 interconvert again to make a new 4-carbon molecule and 6-carbon molecule. The 4-carbon molecule is a precursor for amino acids, while the 6-carbon molecule can be used in glycolysis. The same reversal of steps in option 4 can be here as well. The pentose phosphate pathway takes place in the cytosol of the cell, the same location as glycolysis. The two most important products from this process are ribose 5-phosphate sugar used to make DNA and RNA, and the NADPH molecules which help with building each other molecule.
Hi and welcome for today's vid. Today we're gonna talk about fructose and galactose metabolism. But first, let's look about what is exactly fructose. Fructose, or basically fruit sugar, it is a monosaccharide and it can be found in a lot of different foods such as apples, oranges, kiwis, sugarcane, and honey, and a lot of different more fruits. It is sweeter than sugar, so it will lead to more low calorie dense food. Now let's look into the pathway of fructose. So fructose is converted to fructose 1-phosphate, and the enzyme that catalyzes it is fructokinase. So let's just remember, kinase means a phosphate group gets added. Fructose 1-phosphate can go to two different paths, which is the glyceraldehyde and the HAP, and these two can form glyceraldehyde 3-phosphate, or other known as the G3P, and this is the entryway to glycolysis. How does our body uses or metabolize fructose? So it is metabolized in our liver, which is called fructolysis. It is a process, and basically the liver converts the fructose into energy, and if may natira doon, it will convert to glycogen, and if it is fully stocked, it will convert to triglycerides, or other known as fat. Fruits are really loaded with water, fiber, and other nutrients, which is really essential or healthy for our body. But in a lot of foods that we eat, it must be consumed in moderation. Improper metabolism of fructose can lead to fat buildup and fatty liver disease. And now let us look into galactose metabolism. Galactose is a monosaccharide like fructose. In other words, it is a simple sugar which is normally transformed in the liver before using it as energy. Galactose is also metabolized from lactose. It is produced from milk and other dairy products and intestinal mucosal cells. It is also produced in lysosomal degradation of glycoproteins and glycolipids within the cell-produced galactose. This is the metabolism of galactose in the cytosol. Here, by using or utilizing ATP, form galactose 1-phosphate in the presence of the enzyme called galactokinase. The galactose 1-phosphate reacts with UDP and in exchange to form glucose 1-phosphate and UDP galactose in the presence of the enzyme called galactose 1-phosphate uridyl transferase. UDP galactose on the condensation of glucose gives lactose. UDP galactose also produces glycosaminoglycans, glycoproteins, and cerebrosides and glycolipids. The remaining UDP galactose is now converted to UDP glucose, and this is used for the synthesis of glycogen. Glucose 1-phosphate is converted to glucose 6-phosphate and this enters to glycolysis for the production of glucose. Let's briefly talk about carbs or carbohydrates. Carbohydrates provide our body with glucose, which we can use in our everyday lives us energy so we can play, we can study, we can do a lot of physical stuffs because of the carbs or carbohydrates. Carbohydrates can come in many different forms of food like pizza, bread, and many other stuff. And one of them is sugar or fructose. Fruit sugar is an important source of energy for our body, and galactose is also an essential carbohydrate for cellular metabolism as it contributes to energy production and storage in several human tissues. And some people cannot fully break down galactose, or people that has galactosemia. Galactose makes up one half of lactose. Lactose is a sugar that is found in milk. And people with galactosemia, it can lead to damage in their liver, brain, kidneys, and eyes. And sadly, the only way to treat galactosemia is to eliminate lactose and galactose in our diet. So we should be aware of that if we have galactosemia. Also, some people have fructose intolerance and they cannot digest fructose properly, or there is a lack of an enzyme needed to break down fructose. And typical symptoms include low blood sugar, sweating, confusion, and kidney damage. The diagnosis is made by analyzing a sample of liver tissue, and the treatment for this is avoiding fructose in the diet, and when needed, taking glucose tablets. In learning all of this, we really need to take into more consideration the foods that we eat every day. Eating is really wonderful, to be honest, but we cannot just eat everything we want. We need to eat everything in moderation. We need to pick the food that we eat every day, because not every food is good for us. Not every food is beneficial for our body. So let us examine more and learn more about the substances through foods that we intake and eat every day to have a healthy body.
Glycolipids and glycoproteins have the short fragments of the sugar attached to the proteins of lipids. So, as you can see, the short fragments of the sugar either attach to the proteins or lipids or phospholipids. For example, glycolipids and glycoproteins have a short high-egg carbohydrates or sugar change proteins out of the cell surface. In this structure, you are required to label which one is the external surface. How we know because the role of glycolipids and glycoproteins they are more to the cell right condition, whether we can recognize the cell or not is based on these glycolipids and glycoproteins. Most of them they must be in in the external surface, clear not it's something like I always say that glycolipids and glycoprotein is like our car plates. The number plates of the car. You don't hide them inside your car because you want to recognize your car. Let's say I have 10 MTV. Same color, same design. Now, how you know which one is your car? You will look for these car plates, right? So, inside the cell, also the something how we recognize them based on these glycolipids and glycoproteins. Because of there are four recognitions means that the one must be external surface. Why this is the external so this one is the external surface because of the presence of the glycolipids and glycoprotein. So due to the presence of oligosaccharides or short fragments of sugar of the glycoproteins and glycolipids indicates that the marked region is the external surface of the membrane.
Gluconeogenesis is the formation of glucose from non-carbohydrate sources. Carbohydrates are broken down in human body enzymes into glucose, but it is just a breakdown. In the special circumstances, substances like fat, amino acid, pyruvates are converted into glucose. That is the production of glucose from new sources. This gluconeogenesis can be defined as a synthesis of glucose from non-carbohydrate sources. Our brain, red blood cells, kidney, cornea or lens, and blood cells are need continuous supply of glucose to remain functional. So we have two important processes, the glycogenolysis, which is the breakdown, and gluconeogenesis, which is the production of new glucose. So, the major source of glucose are the exogenous glucose, that's the glucose contained from the food we eat, namely carbohydrates. Galactose and fructose can be converted into glucose in the liver. And now, this exogenous supply is rapidly used up with the body fasting around 4 to 5 hours on average during fasting. The glucose supply provided by the breakdown on glycogen deliver. It can last somewhere between 10 to 18 hours. Glycogen breakdown drops dramatic after the first 12 hours during glycogenesis and increases. After say 24 to 48 hours, glycogenesis represents the own source of glucose. During an overnight fast, 90% of glucose production occurs in livers, while in the remaining 10% in the kidney. But in the prolonged fast night, kidneys became the major glucose-producing organs.
What is carbohydrates? Carbohydrates are the most common class of biochemical compounds. The basic building block of carbohydrates is the monosaccharides, which consists of 6 carbon atoms. Sugars are sweet, short-chain, soluble carbohydrates that are found in many foods and supply us with energy. Simple sugars are such as glucose consist of just one monosaccharides, while complex carbohydrates or polysaccharides consist of thousands hundreds or even thousands of monosaccharides. They include starch, glycogen, cellulose, and chitin. They generally either store energy or form structures such as cell walls in living things. Health, health benefits of carbohydrates. It gives us energy for the body, helps to sleep better, helps to prevent diseases, useful to control weight, helps to keep memory sharp, uplifts mood, provides fibers to the body, prevents our blood clots, improves the digestive system, and best nutrients for the athletes.